The importance of a stable, safe, loving, caring home cannot be overemphasized as a means to foster the positive potential of the epigenome during childhood.
It's one of my favorite passages in the book, but I think it speaks directly to what you were talking about in relation to epigenetics. And I believe it's suggesting that lifestyle changes really do have an effect upon our genetics intergenerationally.
Let me say that it is more than an honor and a privilege to introduce this man. Ralph is the reason that I am sitting where I am, and he has been a friend and a mentor in many ways to me, and for that I am eternally grateful. So it is my great pleasure to introduce Ralph Zehr. And Ralph is going to speak with us today on the topic of dynamic developments in epigenetics. Ralph, the platform is yours.
Well, thank you, Marjorie, very much for that kind introduction. It's really been a pleasure to work on this project, and especially now to see how beautifully it's blossoming under Marjorie's direction. Welcome to this lecture on dynamic developments in epigenetics. We will start this presentation with the definition of epigenetics. We will briefly review genetics to provide a better understanding of how epigenetics exercises control of genetics and consider how our emerging understanding of epigenetics has changed the perception of the central dogma of molecular biology. The commonly utilized epigenetic metabolic pathways will be covered, along with the inactivation of the X chromosome in female mammals, which is among the most dramatic events in biological history. Finally, we will consider the significance of neuronal pathways in epigenetics, particularly during childhood development. The companion paper to this presentation covers the etiology and treatment of cancer, as well as additional important epigenetic functions. Epigenetics is the study of how the expression of gene activity is controlled at the cellular level without altering DNA molecular structure. Epi is Greek, meaning above or beyond. In living cells, epigenetics, or the epigenome, exercises significant control over when genes are turned on or off. Mind over matter, or more specifically in this case, mind over body, is a metabolic reality. The Urantia book states, mind is always creative. The mind endowment of an individual is always competent to produce a suitable and serviceable body for the living creature identity. Each of us arrives on this world having received a unique combination of genes from both of our parents in the form of DNA. Early in life, we will begin to recognize the strengths and weaknesses that we've been dealt as we progressively become aware of our personal potentials. Opportunities to demonstrate our abilities, to express our needs and desires, and exercise the strength of our unique personalities will abound as our awareness of our surroundings expands. How we play our hand will have long-term consequences. Now let's examine DNA and RNA, starting with their building blocks, desoxyribose and ribose sugars. Desoxyribose sugar composes part of desoxynucleic acid, DNA, and ribose sugar is part of ribonucleic acid, RNA. Desoxyribose and ribose are both 5-carbon sugars and are nearly identical chemical compounds, differing only by the absence of a single oxygen atom in desoxyribose compared with ribose sugar, hence the name desoxy, meaning the absence of an oxygen atom. The blue highlighting of the remaining H on the desoxyribose sugar molecule marks the missing oxygen atom at C2, whereas with the ribose sugar molecule, the intact hydroxyl group is highlighted in red. One other important difference in the chemical makeup of RNA and DNA is that in RNA, uracil is substituted for thymine. RNA performs many important functions. The most critical is carrying copies of the information stored in DNA, which contains all the instructions for building all the proteins required to produce a complex living organism, such as a human body. Recently, its important role in epigenetics as non-coding RNA genes has emerged. This schematic drawing displays a cross-section of a DNA molecule in a strikingly geometric configuration. The blue rectangles on either side, consisting of the sugar and phosphate molecules, are in reality curved in a double helical configuration. The central section consists of the nucleotide base pairs joined by hydrogen bonds, similar to rungs on a spiral ladder. The 4 nucleotides in DNA always assume a paired configuration in which adenine and thymine are joined by 2 hydrogen bonds, and cytosine and guanine are joined by 3 hydrogen bonds. It should be noted that there are no differential chemical forces between the nucleotides that can determine their sequence or positioning along the DNA molecule. Hydrogen bonds are among the weaker bonds in chemistry, enabling the strands of DNA to readily separate and reunite as occurs during DNA transcription and replication, much like opening and closing a zipper, a process that occurs millions of times each day throughout your body. DNA contains the blueprint for building your body. It determines all the physical features, traits, and characteristics of the body known as your genotype. It provides your physical identity. This color-coded standard chemical display shows the strands are aligned in opposite directions, as noted by the 5' end and the 3' end of the nucleotides. A hydroxyl group is located at the 3' end, whereas a phosphate group is at the 5' end, each with its characteristic different chemical properties. The chemical processes associated with DNA are direction specific. A little known fact concerning Francis Crick, who with James Watson is credited as a co-discoverer of the DNA molecular structure in 1953, is that he believed it was astronomically improbable for DNA to have arisen spontaneously on early Earth. This is known as the theory of panspermia. This image of RNA polymerase II represents what is considered the heart of molecular biology. Following the discovery of the structure of the DNA molecule in 1953, great strides in molecular biology led to the discovery of how DNA synthesizes RNA, which in turn carries the molecular information to ribosomes to produce protein. This image shows RNA polymerase II in purple in molecular detail during transcription. The double-stranded DNA is held in the jaws of the RNA polymerase, in which the two strands, the bright orange and the yellow orange, have been separated or unzipped. The sent strand, bright orange, is actively being transcribed. It is being copied onto the single strand of RNA colored green. This process takes place within the cell nucleus, as indicated by the out of focus strands of coiled DNA, representing their compacted typical appearance when stored as chromatin in the nucleus. When released, the RNA strand will undergo further modification in the nucleus, which will include splicing and reattaching. The matured messenger RNA will then be transported to the ribosome located in the cytoplasm, where the information it carries will be translated and appropriate amino acid molecules will be assembled in precise sequence, producing a new protein molecule. This entire process, which creates the building blocks that compose an entire living organism, became known as the central dogma of molecular biology, which, when simply stated, is the flow of information from DNA to RNA to make a functional product, a protein. We now know, based on epigenetics, that this profoundly simplistic concept does not begin to reflect the true complexity of the host of interacting factors controlling protein synthesis. It has been summarized by Lodish et al., open quote, nucleic acids function as the brains and central nervous system of the cell, while proteins carry out the functions they specify, close quote. Methylation was the first recognized epigenetic metabolic pathway and continues to predominate. Histone modifications by acetylation and methylation is the second most widely recognized metabolic pathway. We now recognize the importance of long non-coding RNA genes, both in terms of epigenetic functionality and their domination of the overall human genome compared with coding genes. The role of phosphorylation and ubiquitination are discussed in the accompanying paper. Methylation involves adding a methyl group circled in red that in almost all cases is added to cytosine at the C5 position. This chemical reaction is performed by the enzyme DNA methyltransferase 1, for short, DNMT1. Methyl groups are frequently placed at the promoter region of the gene, silencing it. The epigenetic effect of methylation, therefore, is usually to turn genes off. A short segment of a DNA molecule displayed in its usual double helical configuration shows two methylated cytosine nucleotides as two white balls. They are located adjacent to one another on opposite strands of the DNA molecule. This is a realistic display of DNA methylation and is at the same time a thing of beauty. De novo and maintenance DNA methylation are different processes requiring different DNA methyltransferases. De novo methylation is performed by enzymes DNMT3A and DNMT3B, whereas maintenance methylation is performed by DNMT1. When the methylated strand of DNA is duplicated, maintenance methylation requires that additional methyl groups be added to the new strand opposite the previously de novo methylated sites of the original strand to maintain the same level of distribution of methylation. De novo methylation is particularly active during early embryonic development of organisms. Active and passive demethylation, indicated in the right lower corner, commonly occurs during spermatogenesis and oogenesis. Folic acid is an essential vitamin belonging to the B complex group. Its functions are producing red blood cells, producing DNA, repairing DNA, and conducting one-carbon folate mediated metabolism. The most common cause of folate deficiency is excessive alcohol intake. Ingested alcohol decreases absorption of folate by the gastrointestinal tract, and at the same time increases clearance of folates through the kidneys, both contributing to folate deficiency. Other causes of reduced GI absorption of folates include Crohn's disease, celiac disease, cancers, tumors, particularly of the GI tract, and kidney dysfunction requiring dialysis. The green box in the left upper corner indicates that the main function of folate-mediated metabolism is supplying S-adenosyl methionine, or SAM for short, that's SAM, circled in red, with the methyl groups required for DNA methylation. It is the major methyl donor in the body, and since methylation is the most important metabolic pathway for epigenetics, it is required for most epigenetic processes. As illustrated here, the folate and methionine cycles are closely intertwined. The green oval representing the enzyme methionine synthase, labeled MS, and its cofactor, vitamin B12, participate in both cycles. In addition to donating the methyl groups for DNA methylation, it also provides for the methylation of RNA, histones, other proteins, and phospholipids. Another closely related folate-mediated metabolic process, not shown, is the synthesis of pyrimidines and purines, both of which are the major components of all nucleotides. Folate deficiency can cause alteration of the cellular environment that could modify or block one-carbon metabolism. In this case, the dietary environment represents a significant potential epigenetic factor. DNA methylation and histone modification are considered together because of the frequent overlapping and interaction between these two epigenetic metabolic pathways. On figure A, we have a schematic display of a short segment of the DNA molecule. The cytosine nucleotides are methylated. The function of messenger RNA is blocked, as illustrated by the X, turning the gene off. In figure B on the right, the upper diagram illustrates histone modification by acetylation of histone tails. There is a row of acetyl groups labeled AC above the histone molecules, represented by the empty circles. 8 histone molecules are joined together, simulating a spool-like structure on which the DNA is wrapped like thread. The addition of acetyl groups to the histone tails reduces the attractive electrical charges between the DNA strand and the histone, which results in loosening of the DNA strands, making them accessible to transcription, turning the gene on. At the bottom right, the deacetylation of histones reverses the above process, returning the genes to their inactive status. An efficient and reliable storage and retrieval system is required for DNA in order to provide access to the approximately 23,000 coding genes, as well as the much larger number of non-coding genes. This process is complicated by the fact that the length of DNA in each cell nucleus averages 1.8 meters, which is completely stored within the nucleus, measuring 2 microns in diameter. DNA is stored as chromatin in the cell nucleus. The epigenome determines whether it is tightly or loosely coiled, which in turn determines accessibility for genetic expression. This illustration displays the continuum starting with the chromosome, continuing as inactive, tightly stored DNA, followed by loosely bound, accessible strands, ending with parallel, separate, unzipped DNA strands, typical of DNA actively undergoing duplication. Histones are an integral part of the DNA storage and retrieval system. Nucleosomes consist of 2 pairs of 4 histone molecules labeled H3, H4, H2A, and H2B for a total of 8 referred to as octamers that functions as a spool on which DNA strands are wound. The strands make 1 2/3 turns on each nucleosome, containing 147 base pairs of nucleotides. Histones H3 and H4 undergo the majority of epigenetic modifications, although histones H2A and H2B also are subject to some alterations. This storage and retrieval system of DNA is highly conserved, indicating that it is widely used throughout the entire spectrum of living organisms and is known as the histone code. Long non-coding RNA genes, defined as those non-coding RNA genes that are greater than 200 base pairs in length, act at nearly every level of gene regulation. The location where these long non-coding RNA activities occur within the cells is divided between the nucleus, which is partially shown on the left, shaded in lavender, in contrast to the cytoplasm shaded in light orange on the right. Their actions involve all three major phases of transcription, pre-transcription, transcription, and post-transcription. During pre-transcription, they act as decoys in which a long non-coding RNA attaches to a protein molecule involved in the initiation of transcription, thus preventing gene function. During transcription, it can interfere directly by transcription modulation, again preventing the gene from being expressed. During post-transcription, it can interfere with messenger RNA stability, with splicing modulation, with editing of the messenger RNA, and prevent cellular distribution. The activities mentioned thus far all occur within the cell nucleus. Additional activities in the cytoplasm include interfering with the messenger RNA stability, causing messenger RNA degradation, and interfering with post-transcriptional activities at the level of the ribosome. All these discoveries are leading up to a revision of the central dogma of molecular biology. Among the areas of epigenetics currently under investigation, the non-coding RNA genes, particularly the long non-coding RNA genes, have attracted the greatest interest. Non-coding genes refer to genes that do not code for proteins, in contrast to the genes referred to by the central dogma that pertain exclusively to sequencing amino acids in protein construction. Long non-coding RNAs occupy an estimated 80 to 85% of the genome and control the coding DNA genes that occupy approximately 2% of the genome. The present list of recognized long non-coding RNAs numbers in the tens of thousands. Its role in cancer diagnosis and treatment has become transformative. The central dogma, with its focus exclusively on coding genes, is under challenge. It presently states genetic information moves from DNA to RNA to protein. In my opinion, the time has come for its revision, which should read non-coding RNAs as expressed through epigenetic molecular pathways controls the flow of genetic information from DNA to RNA to protein. Inactivation of an X chromosome in all female mammals is an essential process. The X chromosome contains approximately 1,000 protein coding genes, while the Y chromosome contains approximately 75 protein coding genes. Females with two X chromosomes would be subject to excess protein synthesis if there were no compensation. Inactivating X chromosomes in the entire class of mammals is a spectacular achievement. In most cases, an inactive or non-functional chromosome is incompatible with life. In this case, inactivation is essential for life. This is known as dosage compensation and rebalances the relative gene activities between males and females. It is accomplished during the first 6.5 days following fertilization in humans and is completed by approximately the 90-cell zygote stage. The selection of whether the paternal or maternal X chromosome will be silenced is random, and once complete, it will be passed on to each daughter cell as the zygote continues to grow. The tortoiseshell cat pictured here, also commonly known as a calico cat, is a visual illustration of inactivation. This cat has a mixture of black and orange patches. This pattern was first recognized in laboratory mice by Mary Lyon, a prominent geneticist in the late 1950s. She knew that the fur color gene was carried by X chromosomes and concluded that the mottled coat color was a result of silencing an opposing fur color gene by X chromosome inactivation. The orange patches result from expression of the gene inherited from one parent, whereas the black patches express the gene fur color from the other parent. Since the process of determining which X chromosome is inactivated is random, the color of hair from each hair follicle is a 50-50 proposition. The bar body is the remnant of the inactivated X chromosome. It appears as a dense, darkly stained spot on the periphery of each somatic cell nucleus in the human female. It contains a large amount of heterochromatin, which causes X chromosome inactivation. Identification of the bar body is a common legally accepted confirmation of biological sex. If you sample tissue from the bar body and stained it with hematoxylin and eosin stain, a widely used staining technique for microscopic examination of tissues, it would appear similar to the left side of this image. The darker oval-shaped structures are cell nuclei that are crowded together, characteristic of heterochromatin. The euchromatic tissue on the right displays a normal distribution of nuclei. Deacetylation with heterochromatin deposition permanently reduces gene expression. It is the major cause of inactivating the X chromosome. On Wednesday, April 5, AD 30, Jesus advised John Mark, who had become a serious youthful follower, open quote, "Your whole lifetime will be more happy and dependable because you spent your first 8 years in a normal and well-regulated home. You possess a strong and well-knit character because you grew up in a home where love prevailed and wisdom reigned. Such a childhood training produces a type of loyalty which assures me that you will go through with the course you have begun. The biological life of the fetus begins with fertilization of the ovum, at which time the haploid genetic factors located in the ovum and sperm become fused to form the zygote, establishing the full diploid complement of DNA that together will determine the new individual's genotype. The personal life of the child will begin at birth, which combined with all the environmental and epigenetic factors will significantly further influence continued development. We will now consider the scientific information that strongly promotes child welfare based on our recently expanded understanding of the importance of the epigenome during the formative period of childhood. Brain growth and maturation is a complex process beginning early during fetal development as the organ systems of the body are being laid down. Brain development undergoes 4 major stages: neurogenesis, neuronal migration, myelination, and synaptogenesis. During neurogenesis, neurons and other supportive cells appear. This is followed by neuronal migration, in which neurons within the brain relocate and are rearranged. Myelination is the process of coating the nerve tracts with myelin, an insulating substance essential for nerve signal transmission. During synaptogenesis, neural pathways are established and connections between the various individual neurons and brain components are matured. In most mammals, these processes are largely complete at the time of birth, whereas in primates, it is much more protracted and in humans extends to early puberty and beyond. Serial MRI brain scans performed on children have demonstrated significant variation in rates of growth of different regions of the brain. For example, during early childhood, there is relatively rapid expansion of the gray matter shortly after birth, which then begins to recede. Other areas of the brain also demonstrate variations in growth rates over time. Scientific data establishes a functional relationship between neural stimuli and connectivity within the brain, indicating a strong environmental influence on brain growth and function. The human brain is by far our most environmentally responsive organ, as evidenced by the individual creativity and the ability to react decisively to environmental threats. No other organ has access to the extensive sensory input and the capacity to respond effectively as does the brain. A bad environment can very effectively spoil an excellent inheritance, at least during the younger years of life. Good social environment and proper education are indispensable soil and atmosphere for getting the most out of a good inheritance. Environmental epigenetic factors can be both positive and negative. Rich learning experiences can permanently affect brain function in a young child by associating learning with a pleasant and positive experience, resulting in the child recognizing lifelong learning as both pleasant and deeply satisfying. Positive experiences can result in beneficial lifetime changes in a person's mental attitude and outlook. Negative experiences can also establish lifelong negative attitudes. There is scientific evidence based on carefully established animal models, primarily rats and mice, indicating that childhood abuse can have significant lifelong negative effects, which can be passed on to subsequent generations. Children exposed to severe conflict and other highly disturbing experiences, such as abusive situations, can acquire epigenetic changes that permanently alter the receptivity of the neural centers in the brain that control the level of stress hormone, cortisol. This is responsible for signaling the fight or flight response. However, if there is continuous exposure to frequent and regular violence, the brain receptors can become permanently modified, causing prolonged elevated levels of cortisol, resulting in a pattern of recurrent overreaction to stress. The importance of a stable, safe, loving, caring home and community environments saturated with quality adult-child interaction, which serves to stimulate learning and creativity, cannot be overemphasized as a means to foster the positive potential of the epigenome during childhood. Epigenetics is among the most dynamic fields of molecular biology. The time for revision of the central dogma has arrived. The relative proportion of the human genome occupied by non-coding RNA genes, many of which are known to be primarily concerned with epigenetic functions, is far greater than the portion of coding genes. Epigenetics plays a major role in the etiology, prognosis, and treatment of cancer. The recognition of the importance of the CpG island methylator phenotype, CIMP in short, status in virtually all cancers is a major step forward in focusing cancer research on the molecular basis for all malignancies. This topic is covered in greater detail in the paper. Epigenetics can exert a powerful control over genetics, as illustrated by the near complete inactivation of an entire chromosome, the X chromosome, in all female mammals. Jack P. Schoenhoff et al. Reported, the new scientific research shows that environmental influences can actually affect whether and how genes are expressed. Thus, the old ideas that genes are set in stone or that they alone determine development have been disproven. If this is true, and I am personally convinced it is, by extrapolation, it is applicable not only to children, but to humans of all ages. And when one factors in the transgenerational potential of epigenetics, the impact on societal improvement is unlimited. In closing, I wish to point out, in the form of a series of questions, several scientific dilemmas too obvious to ignore that are inconsistent with the prevailing theories of causation based on mathematical probability. For example, what causes the non-random distribution of CP islands throughout mammalian genomes? Next, what is the explanation of the puzzling inconsistencies exhibited by methyltransferase activities? What is the origin of the design and structure of the DNA molecule that is phenomenal in so many ways, of which Francis Crick was keenly aware? How were the design and the instructions derived for the thousands of complex proteins exemplified by RNA polymerase II that performs copying of DNA onto RNA at incredible speeds, essentially flawlessly, providing the precise information to assemble amino acids in exact consequences for thousands of proteins in each living cell? How could the concept of an information molecule result from an entirely mechanistic molecular process? How was the universal genetic code conceived? How did the universal genetic code originate at the onset of the evolutionary cycle, since we now know it has directed the process from the beginning? Once again, as scientists, we find ourselves face to face with dilemmas for which there appear to be no reasonable, scientifically supported answer or resolution. In such cases, we must reach beyond science for higher level universe truth. The best resource of which I am aware is the Urantia book. The following statements are not scientific statements, but they do provide explanations for the above dilemmas. I quote, "We organized and initiated the original life patterns of this world and planted them in the hospitable waters of the realm. All planetary life had its origin in our three original, identical, and simultaneous marine life implantations. This metamorphosis, transition from vegetable to animal life, took place in the shallow waters of the sheltered tropic bays and lagoons of the extensive shorelines of the separating continents. And this development, all of which was inherent in the original life patterns, came about gradually. Thank you very much for your attention."
Ralph, that was an excellent presentation. We're going to ask Ralph to come forward, and he'll be here for us to have a discussion about his wonderful presentation. As always, Ralph, you wow us with your scholarship and how much great information you can explain to us on a level that we can understand it. So we certainly appreciate your scholarship. What drew you? Was it because of the cancer? Was that what drew you to the study of epigenetics, Ralph? Was that the thing that really got you?
Well, actually, Marjorie, I spoke with a scientist a number of years ago, Urantia book reader, who mentioned that we're told that Jesus' inheritance was average, that his parents were representative of the average human being, so to speak, at that time. But yet his performance, his influence, his ability to bring about positive changes on this planet were astronomically beyond the average. And he said, I believe that this probably represents the results of epigenetics. And that really piqued my interest in this topic. And the more I've studied, the more I've learned about it, the more convinced I am that his observation was right on.
Okay, I think Gard has a question.
Ralph, well done. Thank you so much. My question, you didn't spend much time with it, and I would like you to amplify, please, how lifestyle choices. I know the Urantia University Institute is instituting a new program around lifestyle choices and spirituality. But lifestyle choices, including spirituality, how does that relate to epigenetics? And if you could be specific, and also talk about, I didn't hear anything about telomeres. I'd like to hear a little bit about telomeres and how epigenetics affects our telomeres.
Gard, you asked me challenging questions. First off, certainly from a scientific point of view, we really don't have the tools at this time, or we don't recognize the tools which would enable us to clearly track the epigenetic effects of life decisions, right? Our choices. The study that I referred to, done by Dr. Schoenkoff, was done at Harvard, in which they really attempted to approach this problem, and they did come up with very definite scientific evidence, empirical evidence, that trauma during childhood can produce permanent changes in the brain and actually can result in changes that almost certainly can be transmitted then transgenerationally. And that's one of the big questions at present. And I dealt with this to some degree in the paper, but you're right, I didn't really get into too much here. Now, telomeres, I didn't really get into, and I'm, to be honest with you, Gard, I'm not really aware of the exact epigenetic effects that has.
So I would share with you, there actually has been some great studies. One was called, a book called Blue Zones, which studied populations in Okinawa, Loma Linda, California, Greece, other places where lifestyle choices really changed intergenerationally the epigenetics of those populations. And they compared them to red zones. And red zones would be like New York City, places where there's high stress, Tokyo, and shorter lifespans. And so I would recommend to the audience to read a book called Hacking Darwin. And again, there's been a lot of good research done about telomeres, which extends our lifespan. And as you know, Ralph, in the book, it talks about how our lifespans are intended, if we'd had the proper genetic infusion, to be closer to 200 or 300 years.
Mm-hmm.
And telomeres are actually a part of that, and epigenetic or lifestyle choices. And I do believe, in reference to Jesus, that his own contemplative practices were epigenetically informing, if you will, the expression of his life. So thank you again for your wonderful contribution.
Thanks, Gard. Okay, David has a question.
Yeah, well done, Ralph. You mentioned about the changes that can take place if a child undergoes a lot of stress and difficulties in early life. You mentioned 1 to 8, but what if a person has had a good childhood with good conversation, love in the family, education, but the violence or the the difficulties happen maybe in the teenage years or even later. Do you know if there's research about that, whether that is something that could also change to where it would pass on to other generations or affect a person's, life? That's one question about that. And is there anything that can be done to reverse this?
I'm not aware of specific scientific data, but certainly it's reasonable, at least in my opinion, that these scientifically proved difficulties that become permanent during childhood, I think the data can be extrapolated to at any age, at any stage in life. And for example, we know we're very much aware of the trauma that has occurred among our military personnel and the long-term effects that has. So there's no question but what trauma probably at any stage in life can have long-term effects that are extremely difficult to overcome, as we all know. And but that's a bit that's not scientifically documented to my knowledge in terms of the exact metabolic pathway that occur. We are learning that the neural pathways, which I mentioned at the end of my talk, are extremely important as epigenetic functional pathways.
there have been studies done on the children of Vietnam veterans.
Mhm.
They were mainly looking, of course, at the change in their effects on their genome due to things like Agent Orange. But now, since they have that database, they are going back and looking at more subtle epigenetic changes. And so I think that data will be rolling out. But also the study that is being done at the National Institute of Health called All of Us, where they're trying to recruit 1 million people to give all of their health information over a long period of time, is looking at things like epigenetic changes. So people are looking for these answers, and there's way. I'll make another comment. If you really want a quick deep dive into telomeres, one of the premier telomere people in the world is, her name has escaped me, but she did a spectacular TED Talk about 4 or 5 years ago, but it gets you into the biology of telomeres very quickly. And I can highly recommend that as a quick, accessible thing for people. So there's a lot of information and a lot of interest. I'll say this, that females, while you're in your childbearing years, anything that affects you in any way, like severe stress or trauma or whatever, has the potential to pass on in your genome. And so even though your eggs are supposedly set, you have to remember that gestation happens. And that's the reason this thing about, doing pregnancy, having very high, like the women in Ukraine that were having their babies were pregnant during this horrible time in their country's history. Those children, you have to assume now, we know that they're going to sustain some damage probably to their genome. So anyway, I don't want to take, this is just a subject that's very dear to my heart. So anyway, and we do have a lot of questions from the audience, and Phil has got his hand up. So Phil, I'm going to let you, and then I'm going to do a few audience questions. So please go ahead.
Well, thank you. And thank you, Ralph, for explaining so much about epigenetics that I had no idea about. Is the entire process physical chemistry, or is there mind direction in there somewhere that is known?
Well, I did mention that the neuronal pathways are becoming much more recognized from a scientific point of view as pathways for epigenetic effects, for epigenetic expression. And that's fairly new, and that's much more difficult to really document than to follow the metabolic pathways, which we know so much about. And yet we're even there, we're just scratching the surface with the long non-coding RNA genes, which have really come to the fore in recent years. And as I mentioned, is challenging the basic concept of the central dogma. But back to the question, or back to the comment that Marjorie made about the importance of the gestational stage of development of the fetus. There's a great deal of information related to intrauterine epigenetic effects, and these are well documented. For example, there's a major epigenetic factor involved in the present overweight issue in this country. It's really an epidemic of obesity that we're dealing with. And it's not just in this country, it's really worldwide. And there's clearly intrauterine epigenetic effects that occur that result in infants also becoming overweight even during early development. So that's contributing to the epidemic that we're experiencing worldwide. Similarly, for diabetes type 2, there's a clear-cut, well-documented epigenetic effect that has to do with the even during gestational diabetes. Of course, the mother has higher than normal sugar levels circulating in the bloodstream, which are passed across the placenta. However, the insulin response to that she would normally produce is not transported, it does not cross the placenta, so that the fetus pancreas is overstimulated to respond to this elevated glucose, blood glucose level that occurs, gestationally. So those are two examples of clearly documented gestational epigenetic factors.
Ralph, we have a question from Andrew Froelich in our audience. Are there any studies on the effects of regular meditation on epigenetic processes?
There actually have been a lot of studies on the neuroplasticity and the effect of meditation upon the neural pathways and connected to our epigenetics.
Yeah, that I would ratify that. There are studies at Harvard under Herbert Benson and others that show a direct epigenetic impact from meditation, even within the same small period of time after meditation.
Conrad's had his hand up for a little while.
Well, from what you said, Ralph, it's clear that epigenetic plays an important role in the younger ages of people. But when people are meditating or modifying their outlook on life, when they change their mental attitude, it may have some psychosomatic effects. There are research that indicates that sometimes when certain emotions, positive feelings or emotions are maintained, it affects the hypothalamus, which in turns will trigger the productions of very complex hormones that has beneficial effects throughout the organisms, and sometimes may contribute to bring some cancers into remission. So all those psychosomatic effects seems to indicate that there may be some epigenetic changes that could be triggered at the later ages throughout the life of people. And for instance, if you look at a glass of water, you can have positive feelings by realizing that it's half full, or have negative feelings realizing that it's half empty. The glass won't change, but the reactions within our system will be quite different, whether we maintain a positive outlook or a negative outlook. And I'm just wondering if all those feelings, emotions, and spiritual phenomena is taking place when you're worshiping or praying or meditating or contemplating, it all has a kind of cascading effect that may translate into many faceted chemical reactions in the body. So is it possible that at an adult age or at an old age, you can trigger some changes that will have epigenetic effects and that could be transmissible?
Oh, I believe that's true. And I don't think there's any doubt about positive mental attitude, contributing physiologically to our sense of well-being, for example. Certainly, the Urantia book places great emphasis on the positive effects of our desires, on our sense of what we want to achieve, of our purpose, the importance of our sense of purpose on all of life. And in fact, from a spiritual point of view, what we desire to achieve is really considered more significant than what we really have achieved. It's the first step in transmuting potential to actual. And that's an ongoing process that will follow us forever, I believe.
Taken from the Urantia book, one of the things that I realized our thought adjusters, when we have some thoughts that are based on values, our thought adjusters are transmuting those thoughts, and it takes shapes into as the soul. And I'm sometimes wondering if the soul, as it's developing, may have some impact on our feelings, our intuitions, our a sense of well-being, so to speak, and that may in turn have epigenetic effects. And I mean, that's an interesting line of thought too.
Yeah, the book talks, deals directly with soul consciousness and the importance of soul consciousness, the results that it can have, and in fact, we'll be doing a conference in 2024 at Baltimore on the soul, and this will be one of the big topics that will be discussed there.
Byron's had his hand up for a while, so please, Byron.
Thanks so much for this talk. I really, really appreciate it. Wanted to ask you whether you think we've come full circle and we have to become Lamarckians, if you know what I mean. That Lamarck, who was rejected early, was a rival of Darwin, that experience affects the genome or the epigenome.
Well, I think we are really, that's becoming widely recognized now, particularly based on the study I mentioned by the Harvard group. I think we're becoming much more aware, as a number of you have pointed out, of the psychosomatic aspects of it all.
We have a really thoughtful question from Nian Emerson Chase, who has asked, in thinking about this, what are your thoughts about the mind and spirit gravity circuits of the infinite spirit and eternal son possibly being related to our genetics?
I think we're only beginning to recognize, at least at the scientific level, the importance of the neural connections that control, our physiology, and I'm not sure we can really make any scientific comments on that.
All right. Anybody else have a comment?
It's not coming from our laboratories, Ralph, but I think it's coming from a revelation, which is good enough for me. It is to the mind of perfect poise housed in a body of clean habits, stabilized neural energies, and balanced chemical function. When the physical, mental, and spiritual powers are in triune harmony of development, that a maximum of light and truth can be imparted with a minimum of temporal danger or risk to the welfare of such a being. It's one of my favorite passages in the book, but I think it speaks directly to what you were talking about in relation to epigenetics. And I believe it's suggesting that lifestyle changes, like is shared in Blue Zones, the book Blue Zones, really do have an effect upon our genetics intergenerationally.
Oh, that's a marvelous reference, and it has so many practical ramifications about avoiding toxins, for example, avoiding all kinds of chemical toxins, and mental toxins. And one of the big ones is impatience, the book says, is a spiritual poison. And that implies that it can actually poison our spiritual functioning and can interfere significantly with it. Yes, the book is loaded with good mental hygienic advice and physiological hygienic advice. And it clearly provides goals for our developing, like I said, good mental hygiene.
Yeah, there is a book I would recommend to the listening audience. Some of you may have read it, by Oprah Winfrey and Bruce Perry, who's a neuroscientist from the University of Chicago. And it's called What Happened to You? And it's really one of the best books I've read on trauma. And I've been reading a lot over the last 2 years during the pandemic. But Bruce Perry really nails it. And he says, to the question earlier, that when trauma occurs in early childhood in the womb and or during the first few years, that is the most difficult trauma to heal. It is powerfully embedded in the reptilian brainstem, and it requires a great deal of work. So there is a difference between trauma that occurs in those early years and trauma that occurs later in our life. I really recommend that book because Oprah gives the vignettes from 40 years of shows and Bruce Perry gives the science. So
this is from Andre Redatus. What are the 1, 2, or 3 most significant physical, emotional, or spiritual environmental factors to avoid in the development of the child?
Yes. I think one that we're just beginning to recognize is during childhood, the problems that result from sexual abuse of young children. Those who have worked with these children have observed that they have many serious psychological issues to deal with that have to do particularly with their sense of well-being, their sense of their after all, their body is very much occupies a great deal of all of their psychic activities during early childhood. And when that's violated, that permanently changes their sense of well-being, and it's just it's extremely difficult to overcome in later years.
there is a great test called the ACE test, which I recommend people look at. It's adverse childhood experiences. And that, Andre, lays out the kind of environmental threats and risks that are most prominently in the minds of psychologists and neuroscientists.
And he is asking the antithesis to that, or what are 1, 2, or 3 of the most significant physical, emotional, or spiritual environmental factors to proactively ensure the development of the child? And I think your quote about John Mark was one of them. That's my personal one, but please answer, Ralph.
I'm at the present time in the stage that my grandchildren are young adults, and I would say my experience in fathering young children involved exposing them to as much as possible to the pleasures of learning, of providing wide experiences that are gained through travel, and of course, good entertainment. There's a lot of good entertainment out there. Appreciation of music, appreciation of art, and just exposure to other people, to other races, to other cultures. And we did a great deal of that. At least we tried. I think a broad exposure is probably the best education we can offer young children.
I've been involved in a project that GARD is aware of in my old home state of Alabama, where we were trying to work at securing the health of children, because there came across to us a demographic that is pretty worldwide. And that is that if you have not secured a child's mental, physical, and spiritual health by the time they are 5 years old,
Mhm.
at the very best you can expect is that they will be a zero to society. The majority of them will become they will not be an asset. In other words, you're not, and so it comes down to even a socioeconomic thing about investing in our children while they are still so young, because they can secure so incredible damage at these very young ages. And so it behooves all of us, whether we have children or not, or we're older or whatever, to work at securing the health for all children, because they are so incredibly vulnerable, more so than we've ever realized. And we are realizing it more and more every day. Sharon Holland has asked, do studies show what happens to a child reared mostly in a stable and well-regulated home with infrequent traumatic experiences?
Well, I did make reference to the Harvard study once again that did clearly document that changes occur in trauma brain centers that are involved with the excretion of cortisol, which, as you all know, is associated with the normal physiological response to severe trauma and to severe threatening environmental situations. And this is based on animal studies. Obviously, we can't do human studies on this sort of thing, but the animal studies make it clear that permanent changes result in the brain centers, which will then continue to overreact, as it were, to threatening situations. And this will cause a overreaction as a habitual response to any form of trauma. And it will just be an inappropriate physiological response.
Marjorie, I might share a book.
Oh yeah, that's a great one. Yeah.
Lisa Miller, who's actually done the studies that shows that spirituality, and she was shunned by many of her associates who sort of have that traditional dogma. Spirituality, are you kidding? But spirituality is the flip side of trauma and depression and mental health issues for children. And it's a really great read. I recommend it.
We do have a question that's in that vein. Do you think worship can modify our epigenetics and in the long run perform the function of that Adamic genetic uplift that could not be completed?
Well, that's a fascinating, speculative question. And we don't really know what exactly we've missed as a result of the failure of having inherited a greater amount of the violet blood. But again, I don't doubt at all that we can clearly modify our our outlook, we can, and this is something that we can control. This is clearly within the realm of choice. When we choose to follow the more spiritual pathways, choose to follow, as you pointed out, to take advantage of the effects of meditation and of seeking greater spiritual guidance for choosing and actively, proactively seeking greater input from our thought adjusters and the spirit of truth, who's always there waiting, hoping to provide us greater insights, greater spiritual understanding, greater peace, and greater love, and greater caring.
Thanks, Ralph. Conrad's had his hand up again, so please, Conrad.
What I find interesting in Andre's questions and your answer, Ralph, to that question and what you just said, is all this seems to be boiling down to the cultivation of the three supreme values: beauty, goodness, and truth. And I noticed, though, that people who have a kind of sacred flame, the passion in life for some causes, I see that those causes are often, almost always, related to beauty through arts, or goodness through their dedication to all sorts of causes to help for the betterment of the world, or truth. They're passionate about finding new truths. And in the Urantia book, you mentioned that the main activity of worlds settled in life has to do with the further advance of truth, beauty, and goodness. So it seems that the conditions that would favor those epigenetic developments would be well associated with those values, because they trigger within us so many changes.
Oh, I think you're absolutely right. I mean, goodness, beauty, and truth are the avenues through which we see God, and that's the way He speaks to us, and we're surrounded with it all the time. The beauties of nature, the beauties of just the sounds of the birds, and the aromas of the flowers, and there's just so much environmental input, and we know that epigenetics is largely a response to environmental factors, and there's just it's really pretty simple that goodness, beauty, and truth is where we're at. It's really that's the sort of where the tire meets the road in terms of our understanding of God, and our sense of God consciousness is very much a reflection.
And to me, it's a kind of litmus test. If we look at the medias, the negative tendencies or the negative focus of practically every media in today's world, that may have a lot of negative effects in the feelings and the minds entertained in people's. And with the pandemics, people were exposed to medias where they were exposed to a lot of negative information, which tended to accentuate those problems. But the antidote to all the negativism that we may have in society may lie precisely in that cultivation of beauty, goodness, and truth.
Thank you, Conrad. You kind of answered this question, Ralph, but Andre was interested in the silencing of one of the X chromosomes in all female mammals. And I think you said that which X chromosome is silenced and which one is expressed is random. In other words, whether you get it from your male line or your female line. Are there any studies that are ongoing that are looking at preferences? In other words, do we tend to be more economically, or we favor the perpetuation of the female X chromosome, like the ones that come down from our mother, than we do from the ones that we come down from our father. Are you aware? Is anybody really looking at that?
I think that's being looked at. It's a fascinating, it's fairly recently come to the attention of geneticists and biologists. And like it was, it was just in the 50s when Mary Lyon, in the late 50s, that she recognized this phenomena of X chromosome inactivation. The studies, the papers I read did not get into detail as to exactly what physiological mechanisms, what molecular machines were involved in this selection of which of the X chromosomes gets inactivated, and that occurs, as I said, randomly in each of the cells existent in the zygote at that stage, which is usually by the 90 cell zygote stage. So I can't really answer your question, but work is being done on that. The only information I could find was that it was considered to be a random actually process.
There was someone in the audience sort of keyed into the word, but we were all talking about neuroplasticity, which is, of course, a big area of science at the moment. In your studies and reading papers, did you find that so many of the majority of the epigenetic things we're looking at, neuroplasticity is where so many of the epigenetic changes were occurring, particularly in children?
Yes, neuroplasticity, as you point out, is a general term, and it involves all of the billions of neurons that are functioning regularly with us. I'm not sure that I have anything specific to add to that.
So Mark Hansen, in his book Buddha's Brain, does an extensive piece on neuroplasticity, and the mantra with neuroplasticity is, as the neurons fire, so they wire. So if you're firing with anger, if you're firing with negative emotions, you're creating a kind of a super highway of awareness that is laid down by anger and fear and anxiety. So the beautiful thing about meditation is that we can grow new neurons and we can repave, if you will, a difficult highway, even in an old age. As Byron pointed out, even a few minutes of meditation makes a huge difference.
A lot of work has been done with early childhood brain stimulation among various genetic issues, genetic diseases associated with some of the childhood disabilities that have resulted from mutations, and stimulation, just visual stimulation, for example, is very important during the very first few days of life, and children that are not exposed to stimulating environments just fail to develop, like you said, fail to develop these certain neural pathways that are so essential.
Thanks, Ralph. Conrad gave us a very excellent quote. Health, sanity, and happiness are integrations of truth, beauty, and goodness as they are blended in human experience. So I'm going to thank Ralph for his absolutely wonderful presentation. Ralph, it was really great, and it's a joy to always be in a conversation with you. I always learn a lot.